Hello students, myself Dr. Shantanu Bandyopadhyay from APJ Kalam Technical University, Lucknow, and today I am going to take the lecture on the topic that is product. Under this topic we will be learning the various concepts, and the contents are: initially we will study about the various introduction parts, the concept of product, classification of products, functional groups which are amenable to the product design, and application of products, and finally we have the concluding remarks for the whole content. Starting with the introduction section, high throughput screening, or what is termed as HTS, and combinatorial chemistry. The application of modern discovery technologies can produce novel lead structures with high pharmacological potency. However, the biopharmaceutical and physicochemical facets of the preliminary leads have frequently been neglected. This often leads to the drug candidates with poor drug-like properties, which face significant problems of late in the drug development. The term product was introduced by the scientist Adrian Albert in 1958, and it relates to the inert derivatives of the drug molecules which undergo various kinds of enzymatic and or chemical conversion in vivo to release the active parent drug. The development of products, which is essentially the chemically modified versions of the pharmacology active agent undergoing transformation in vivo to release the active drug, is now an outright confirmed tool for improving the various properties, viz. Physicochemical, biopharmaceutical, or pharmacokinetics of pharmacologically potent compounds, which enhances the formulation capacity and increases the usefulness of a potential drug. The products provides various opportunities to overcome the barriers during drug formulation and delivery, such as poor aqueous solubility, chemical instability, insufficient oral absorption, rapid pre-systemic metabolism, brain penetration issues, toxicity, and lastly the local irritation. The products also have the tendency to improve the drug targeting, and when applied to an already, it may represent a life cycle management opportunity. Now, what is the concept of product? The basic aim of the product design is to mask the undesirable drug properties such as low solubility in water or lipid membranes, low target selectivity, chemical instability, undesirable taste, irritation, or pain after the local administration, and lastly the pre-systematic metabolism and toxicity. In general, the local basis of use of the products is to achieve the optimum levels of AD/MET, or what is in detail termed as absorption, distribution, metabolism, excretion, and toxicity of the parent drug. Basically, there are three overlapping objectives in the product research. The first basic property is, or the concept is, pharmaceutical, which improves the solubility, chemical stability, and organoleptic properties. It essentially decreases the irritation and or pain after the local administration, and also reduces the problems which are related to the pharmaceutical technology of the API. The second concept is based on pharmacokinetic parameters, which improves the absorption both by oral as well as non-oral routes, as well as to decrease the pre-systematic metabolism, to improve the time profile, to increase the organ tissue selective delivery of the API. And the third concept is based on pharmacodynamic, which decreases the toxicity and improves the therapeutic index of the drug, and this is basically aimed at to design single chemical entities combining the two drugs, which is also termed as co-drugs strategy. Now coming to the next part, that is classification of products. These are basically two types of products. First one is based on carrier link products, and the second is based on bioprecursor products. In case of carrier link products, the active molecule is temporarily linked to a carrier, which is also known as promoiety, through a bioreversible covalent linkage. Once inside the body, the carrier linked product undergoes biotransformation, thereby releasing the parent drug and the carrier. Ideally, the carrier should be non-immunogenic, easy to synthesize at low cost, stable under the conditions of product administration, and undergo biodegradation to non-active metabolites. As you can see on the screen, that the figure depicts how the in vivo bioactivation of the product is carried out using the enzymatic or the chemical transformations. Sometimes what happens in case of product, two pharmacologically active drugs are coupled together into a single molecule in such a way that each drug can act as a promoiety for the other. Such derivatives are termed as co-drugs. Examples include sulfapyridine 5-amino-salicylic acid or indomethacin with paracetamol. Now there is a term which is coined as bioprecursors, and these bioprecursors do not contain a promoiety, and it is produced by the molecular modification of the active compound itself. The bioprecursor product is transformed metabolically or chemically by the hydration process like lactones into statins, oxidations like navubiton, or reduction process like salindac to the biologically active compounds. Based on the site of conversion into the pharmacological active compounds, the products can be additionally classified into two groups. Under the first group, that is termed as type 1, which includes the drugs which are metabolized intracellularly, and the type 1A product example is acyclovir or cyclophosphamide. These are basically metabolized at the cellular targets of their therapeutic actions. Under the type 1B example like carbamazine or captopril, these are converted to the parent drugs by the metabolic tissues, namely by the liver. Under the second category, that is type 2, under this the products are metabolized extracellularly. Type 2A is in the milieu of GIT, example is loperamide oxide. Type 2B example is aspirin. It is metabolized within the circulatory system and or extracellular fluid compartments. Under the type C, it is the near or inside the therapeutic target of the cells. In order to develop the various products are so designed with the help of functional groups, like the foremost, the design of an appropriate product structure is considered at the early stages of the preclinical development, bearing in mind that it might alter the distribution of the tissue, effectiveness and toxicity of the parent drug. The various important factors that must be studied while designing a product structure includes: the first one is parent drug, the functional groups in the active drug chemical structure which will easily respond to the product derivatization. Second is promoiety. This should ideally be non-toxic and capable of being rapidly excreted from the body. Usually, the choice of promoiety is considered with respect to the various aspects related to the disease state, dose and duration of the therapy. And the last one is parent and the product. It is essentially dependent upon the ADME properties need to be exhaustively understood. Besides this, the degradation byproducts which affect the chemical and physical stability need to be studied. Some of the most common functional groups that are responsive to the product design includes carboxylic, hydroxyl, amines, phosphates and the carbonyl groups. The products typically produced by the modification of these groups includes esters, carbonates, carbamates, amides, phosphates and OXIMES. However, the other uncommon functional groups that have been also investigated, such as thiols, which react in a similar manner to the alcohols and can be derivatized to thioethers and thioesters, as well as the amines, which may be derivatized into imines and bases. Now, as you can see on the screen, there are various examples of functional groups, how they can be appended or for the product design. Among the various examples of functional groups, we will start with the ester products. The features of an ideal product include the following: hydrolysis resistance during absorption, weak or no activity, aqueous solubility, good permeability through the cells, chemical stability at different pH, kinetics that allow release of the parental drug. Among the chemical bonds used to link the parental drug and carrier, esters have proven to be promising due to their ease of hydrolysis both under in vivo and in vitro conditions. Esters are the most commonly used products, as approximately half of all the marketed products are based on esters and are activated by enzymatic hydrolysis. Ester products have the capacity to enhance the lipophilicity, thereby increasing the passive membrane permeability of the water-soluble drugs. Some of the examples using this technology are palmyromycin. It is a naphthoquinone spiroskeletal compound which acts on the thyroredoxin system, is converted into its glycyl ester derivatives, which is more than 7 times more soluble in water and which also helps in countering the high lipophilicity of the parent drug. As you can see on the screen, that palmyromycin has been converted into a product, and how the various conversion has taken place by converting into glycyl ester derivative. And on the arrow we can see that its water solubility has been increased by 7-folds. The major drawback of these ester products are that although they are easily formulated, but challenges lie in the accurate prediction of pharmacokinetic disposition in humans, resulting from differences in specific carboxyl esterase activities in the preclinical species. The next functional group is phosphate esters. Hydroxyl and amine functionalities of poorly water-soluble drugs can be easily derivatized to phosphate ester products, which enhances their aqueous solubility to allow for oral as well as parenteral administration, which is due to the presence of diionic phosphate promoiety. The phosphate products invariably display adequate chemical stability, followed by rapid bioconversion back to the parent drug with the help of phosphatases, which are present at the intestinal brush border or in the liver. The phosphate ester compounds are relatively stable when free in metabolic surroundings because they have a low pKa value between 1 and 2. At a physiological pH of 7 to 7.4, the compounds are permanently deprotonated and therefore negatively charged, but are readily activated upon complexation to various counterions in an enzyme's active site. As you can see on the screen, there is one example of anti-cancer molecule, that is miprocifin phosphate, that how it is converted into with the help of a phosphate functional group. And here we can observe that as soon as the pH is observed, aqueous solubility is observed at pH of 7.4, its solubility is increased up to the 1000 fold. In case of phosphate esters, there is an added advantage over the carboxylic esters that they are typically hydrolyzed at the similar rates in different preclinical species by alkaline phosphatases. Mainly due to the challenges encountered in the development phases, only a few phosphate products for oral administration have reached the market. Usually, the aqueous solubility of the phosphate product is enhanced to such an extent that the passive diffusion through the intestinal membrane is prevented, resulting in the precipitation of the parent drug following the cleavage in the intestinal lumen, which leads to the poor absorption of the parent drug. As you can see on the screen, there is an example of product which is based on parental phosphate formulations, that is phosphonatoin, which is anticonvulsant in nature. Here it has led to the aqueous solubility significantly in higher side. Now the third functional group that can be added for the product design is the carbonates or the carbamates. The presence of either oxygen or nitrogen on both sides of the carbonyl carbon differentiates the carbonates and the carbamates from the ester products. These type of products are more stable towards hydrolysis as compared to the ester products, but at the same time more susceptible to the hydrolysis than amides. The esterases are known to convert these products to the parent drug. There is an example of that product which is termed as Z-melagatran, which is anticoagulant in nature. It has been shown that the oral bioavailability has substantially increased up to 20% as compared to the parent drug. Coming to the next functional group, that is amides as product of carboxylic acid and amines. The amine and carboxyl functionalities of a molecule can be derivatized to amides. Amides are used to a limited extent in the product design owing to the relatively high enzymatic stability in vivo. Carboxyl esterases, peptidases or proteases are the enzyme which help in the bioconversion process. These products substantially enhance the oral absorption by producing the substrates of specific intestinal uptake transporters. On the screen, as you can see, there is an antiviral molecule, valacyclovir, and the parent compound, that is the active compound, is acyclovir. As soon as it is converted as a product of carboxylic acid or amines, the oral bioavailability in comparison to the acyclovir has substantially increased to 54%. The last functional group is oximes as derivatives of ketones, amidines and guanidines. The oximes provide an opportunity to modify the molecules that lacks the hydroxyl, amine or carboxyl functionalities and are derivatives of ketones, amidines and guanidines, for example, ketoximes, amoximes and guanidoxymes. Oximes are hydrolyzed by the various microsomal enzymes, also known as xenobiotic metabolism enzymes. Oximes are used basically to enhance the membrane permeability and absorption of the parent drug. Now coming to the next section, that is applications of product, we can see holistically there are various areas where we can apply the product concept, like improving the aqueous solubility, improving the lipophilicity, improving the oral absorption, site-specific drug delivery, then carrier-mediated absorption, improving the parenteral administration, as well as the improving the topical administration. Now if you look into the individual sections, how we can improve the oral absorption. Like in case of most of the drugs, the bioavailability is controlled by the fraction absorbed and the clearance of the drug. So FA or the fraction absorbed is largely controlled by the physicochemical parameters of the parent or the product, that is its GIT permeability, solubility, dissolution rate and the dose number. FA can also be improved by enhancing the dissolution rate, aqueous solubility or the permeability. In general, some of the product strategies overcome the poor absorption by enhancing the permeability, which is effectively achieved by masking the polar or charged moieties of a poorly permeable parent drug. These products are often carboxylic acid based esters. In case of oral absorption, it is also imperative to understand the physicochemical and biological factors that are limiting the oral bioavailability of a compound before embarking on a product strategy. As it is known that the maximum achievable oral bioavailability, that is Fmax of a parent drug, is a function of three parameters, that is Fa, fraction of the dose that is absorbed after oral administration, Fg, fraction of the dose that escapes intestinal metabolism, Fh is the fraction of the dose that escapes hepatic metabolism. On the screen you can see the example of a product, that is oseltamivir, it is converted by esterases, and therein the oral bioavailability has been increased substantially to 80% in comparison to the parent drug. Now coming to the next application, that is improved aqueous solubility. Approximately 40% of the drug candidates produced from combinatorial screening programs have poor aqueous solubility. That is, they have an aqueous solubility of less than 10 micromolars. The conventional formulation strategies such as particle size reduction, salt formation, complexation, and solubilizing excipients do not provide adequate solubility. In these cases, products offer an alternative tool to overcome the solubility limitations of the poorly soluble drugs when first pass metabolism is low to moderate and not the main case of systemic drugs availability. There are various strategies in order to enhance the solubility profile of a parent drug. Like a parent is a BCS class 2 drug that is having low solubility and high permeability. Second, the high dose oblique solubility ratio, that is the highest targeted parent drug dose, will only dissolve in the volumes much larger than the 250 ml over a pH range of 1 to 7.5. Low or moderate hepatic clearance in rats or preclinical species. Hepatic extraction ratio is less than 0.5. High permeability during in vitro assays such as Caketo cell line studies. Fraction of dose absorbed after oral administration is calculated or measured to be low, that is less than 25%. As you can see on the screen, there is an example of product which is water soluble in nature, that is Salindac. This is a bioprecursor product that does not contain a promoiety, but instead its inactive sulfoxide form is reduced to the active sulfide form after oral administration or the oral absorption. Salindac sulfoxide, the product is about 100 times more water soluble than the pharmacological active sulfide. Also, you can see that once it is incorporated as product, the solubility has been enhanced, especially with in terms of lipophilicity, where the log P is 1.5 to at the pH of 7.4, which essentially provides a more efficient oral absorption. Another application of product is improved lipophilicity. The products are generally used to mask the polar and unionizable groups within a drug molecule with the aim of improving the oral drug delivery. Increasing the drug lipophilicity promotes the membrane permeation and oral absorption. Now within this there is another strategy, that is permeability enhancing. It can be applied when low intestinal permeability of the parent drug is a major barrier to the bioavailability, but not the hepatic metabolism. The parent is a BCS class 3 drug that is high solubility and low permeability. It is having low or moderate clearance in the rat or other preclinical species, and the extraction ratio is also less than 0.5. The low in vitro permeability during the screening assays, and FA is calculated or measured to be low, that is less than 25%. For example, the product of melagatrine, that is zimelgatrine, it is basically a zwitterion, which when converted into product, the bioavailability has substantially increased to 3 to 7%. Another approach or the application is based on carrier mediated absorption. Due to the recent advances in the molecular biology and cloning of nutrient transporters, the products targeted towards specific membrane transporters have been designed having similar structural features that would allow them to be taken up by one of the endogenous transporters present at the intestinal epithelium. For example, the peptide transporters are the most attractive targets for the product design because of their wide distribution throughout the small intestine, which has sufficiently high transport capacity and broad substrate specificity. As you can see on the screen, there is an example of valacyclovir. Now this particular product, it is exploited utilizing the carrier mediated transportation. This amino acid product increased the intestinal permeation of the parent drug by 3 to 10 fold, and the membrane transport was not passive but mediated predominantly by the ti and tri transporter systems expressed in the intestinal epithelial cells. Following their membrane transport, both the products are readily bioconverted back to their parent drugs by intracellular hydrolysis. The next application is based on parenteral administration. The introduction of an ionizable or polar pro-moiety to the parent drug is the most commonly used approach in order to improve the aqueous solubility properties for the parenteral administration. The diionic phosphate group present in the several phosphoric acid esters have been developed as potential water-soluble products for the parenteral administration. As you can see on the screen, there is an example of the product, that is phosphanatoin, which is a phosphate ester product of poorly water-soluble phenytoin for the acute treatment of seizures and can be used for both intravenous and intramuscular administration. In phosphanatoin, a phosphate ester is attached to a weakly acidic, that is pK of 8.3 amine functionality of the phenytoin via an oxymethyl spacer group, leading to a remarkable increase in the aqueous solubility, that is from 20 to 25 microgram per ml to 140 mg per ml. The next application includes the topical administration. Under this, there are two different categories, that is ophthalmic and the dermal. In case of ophthalmic drug delivery, the products were introduced to ophthalmology about 30 years ago when the ocular absorption of adenine was substantially improved by the use of its product, that is dipivalyl adrenaline, or in short it is termed as dipivarin. Dipivarin is a dipivalylic acid diester of adrenaline which penetrates the human cornea 17 times more rapidly than the adrenaline, owing to its 600-fold increase in the lipophilicity at a pH of 7.2 as compared to that of the parental adrenaline. Now the next one is dermal drug category. The unfavorable physicochemical properties of many drug molecules lead to poor permeation across the skin, in particular through the most superficial layer, that is the stratum corneum, which provides high resistance for a topical drug delivery. Either the water and lipid solubilities or a balance of the two are important in the optimization of the drug permeation. These optimal features can often be achieved by products. In case of tazarotene, its active carboxylic acid form is esterified to a more lipophilic ethyl ester which still maintains the adequate water solubility. The tazarotene was effectively and reliably absorbed percutaneously, exerted less skin irritation, and was rapidly converted to tazarotonic acid. The less lipophilic tazarotonic acid subsequently released showed no accumulation in the fats and other tissues, in part due to the reduced systemic half-life of this parent drug, achieved by the introduction of a metabolically labile sulfur group that undergoes rapid oxidative deactivation and thus prevents accumulation in the tissues. Now the last part of application is site-selective drug delivery. An ultimate goal in the drug delivery is that it is site-selective, and this may be the most exciting possibility that products offer. Site selectivity may be achieved in 4 different ways. First, by passively enriching the drug in the organ. Second, by selectively metabolic activation through specific enzymes. Third, through transport mediated delivery, and the last is by antigen targeting. And under this, there are 2 types of targeting. The first one is CNS targeting. Clinically, the CNS is one of the most challenging organs to the target, mainly due to the blood-brain barrier. A clear understanding of the transport mechanisms and the enzymatic activity of the BBB helps in achieving enhanced CNS delivery relative to the other sites of the body. For example, the product of dopamine, that is levodopa, which is a substrate for the neuroamino acid transporter expressed at the BBB. Having entered the brain tissue, levodopa is rapidly converted back to the dopamine, and being a hydrophilic molecule, it is trapped there, enabling its pharmacodynamic effects. Traditionally, to increase the CNS drug concentration, the lipophilicity of the parent drug is increased. For this to be successful, the product must have easy access to the brain tissue. Bioconversion back to the parent drug should be highly site selective, and prolonged retention of the parent drug within the brain tissue should be exhibited. The second is tumor targeting. The basic aim in cancer therapy is to design an explicit inactive product which is selective not only to the tumor cells wherein the active drug may be released without causing any toxicity to the healthy tissues. Increase in the levels of certain enzymes in the cells have been exploited in targeted product tumor delivery. The classic example in case of tumor targeting is capecitabine. It is an orally administered carbamate product of 5-fluorouracil that requires a cascade of 3 enzymes for the bioconversion to the active drug. The intact capecitabine is absorbed from the intestine and undergoes bioconversion in the tumors, thus avoiding any systemic toxicity. The viability of 5-fluorouracil after oral administration of capecitabine is almost 100%, with the Tmax of 5-FU is reached within 1.5 to 2 hours. On the screen, as you can see, that there is a structure of the capecitabine on the extreme left. As soon as it enters into the liver, it is affected by the 2 isozymes, thereby converting into the different structure whereby the carbon dioxide is released. Then finally, as soon as it enters into the tumor part or the liver part, it is again transformed into a different structure. And finally, as soon as it targets the actual tumor, it is the 5-fluorouracil you can see. Now concluding the today's module, the product approach to the drug design is a versatile, powerful method that can be applied to a wide range of drug administration routes and formulations for many types of parent drug molecule. However, for product strategies to be successful analysis of parent drug properties, and the proper identification of the barriers are very crucial. Clinically, the majority of the products are used with the aim of enhancing the drug permeation by increasing the lipophilicity and more recently by improving the water solubility. In summary, the products have become an integral part of the drug design and delivery process as illustrated by the increasing number of approved products and patents. We anticipate that an increased application of rational product approaches at early stages of the drug discovery process by the multidisciplinary teams including medicinal chemists, pharmaceutical chemists, drug metabolism, and pharmacokinetic scientists will lead to the development of a compound with better drug-like properties. Thank you.